about the next experiment which was performed by scientists Hershey and Chase in the year 1952 to prove that DNA is a genetic material. What experiment and how they did, that we will discuss here. Scientists Hershey and Chase perform experiment by using virus bacteriophage. They use virus bacteriophage which infect bacterium, and they prove that DNA is only the material which transfer and which is from one organism to another organism and considered as a genetic material. But here they in order to prove DNA, they first took protein and DNA. They consider both the things, that which material is passing. To prove that, they use protein also, that either protein is there or this material is only and only DNA. For that, they use virus bacteriophage. Bacteriophage, this is virus. And they culture this bacteriophage, virus bacteriophage, they culture in different medium. In one medium, they use only and only sulfur isotopes, and in another medium, they use isotopes of phosphorus. And they culture these viruses in different medium. Now, as they culture these viruses in different medium, those viruses are carrying the isotopes. Once they replicate or they divide, they carry or they shows the isotopes of sulfur and phosphorus in them. Here, sulfur is the component of protein. Sulfur is the component of protein. So, the organization of virus, what is the structure of virus? Virus is like this. Such hexagonal protein shell. This shell is made up of protein. Proteinaceous shell. This is the structure of bacteriophage, virus bacteriophage. This is called as protein capsule. This is stalk. This region is called as stalk. Inside this protein capsule, the genetic material DNA. This is the genetic material of DNA. Here also the spring spiral structure is there, that is the protein. Here end plate. This region it shows end plate. This is called as end plate. These end plate shows small opening, perforation. This is end plate. And these end plate shows a small opening or perforation with leg-like structure. You can call it either leg-like structure also you can call them, or we can call them tail-like structure. Tail-like structure for fixation. So these are the tail-like structure or leg-like structure which will be used by this virus to fix on bacterium. So this is the structure of virus, bacteriophage. So in this organization, what is there? Protein is there as well as DNA, only two things are there. When these bacteriophage cultured in the medium of isotope of sulfur and phosphorus, by the time the protein part, sulfur is the component of protein, and protein part will shows the presence of sulfur isotopes, whereas phosphorus is component of DNA. In DNA, with a nitrogen basis, phosphoric acid is there. So phosphorus is the component of DNA. So phosphorus isotopes will find out in the genetic material, that is the DNA, when cultured in different medium. Now, the experiment performed like that. They infect, once this viruses or bacteriophage has been cultured in different isotopes, now these viruses infect bacterium. So we will take virus which is infected, which is cultured in sulfur medium. This is one of the bacteriophage. This is one of the bacteriophage. Suppose this bacteriophage is cultured in the medium of sulfur. Here another bacteriophage. So here Hershey and Chase, this is the genetic material, they use bacteriophage, they use bacteriophage, one which was cultured in the medium having isotope of sulfur, and another one which was cultured in the medium having isotope of phosphorus. Now allowed to infect bacterium E. coli, allowed to infect bacterium E. coli. When it infect the bacterium E. coli, now after infection, what is the process of this infected, or how this attach with? It attach with simple steps. First is the adsorption. Adsorption means fixing of this bacteriophage or virus with E. coli through this tail region. That is the adsorption. Then injection, inject the material, inject the genetic material. So injection. So adsorption and injection is the commonmost process. Then whatever material get injected, if suppose genetic material, then should be replicate. So that comes under replication. But these two are the important thing what we have to discuss about. So once it get infected by this bacteriophage, E. coli get infected by bacteriophage, after that they took this E. coli, whatever number of E. coli and bacteriophage they used in the experiment, then they separately allow this material or keep this material or put this material in test tube and centrifuge it. They centrifuge this material. When they centrifuge this material, now what will be the result? If suppose this is the test tube, and in this test tube, this material, that is the virus, bacteriophage and bacterium, both are kept with some chemical or enzyme and allow for centrifugation, centrifuge. During centrifugation, centrifuge the material, by the time they find out, when we centrifuge certain mixture, we get two layers, upper layer and lowermost layer. So here they found these two layers, that is supernatant and palate. In palate means the material which remain in the bottom part. In palate region, they found the bacterium. Bacterium remain in the palate region. This bacterium remain in the palate region. And virus is having, the body is made up of protein. So that protein get separate. Protein get separate, and during centrifugation, the proteinaceous part, that is the superficial part, proteinaceous part, it remain in the supernatant region, upper part, because it is lighter in weight. It is very, very light. So this protein shell, protein part remain in the supernatant region. This part is called as supernatant. Supernatant. And this lower is called as the palate. So in palate, all bacterium were present in palate and protein shell were present in supernatant. So in supernatant, they found, in these supernatant, they found here, in the region of supernatant, they found isotopes of sulfur. isotopes of sulfur detected in supernatant, whereas in the region of palette they found isotope of phosphorus, and here in the supernatant region they found isotopes of phosphorus inside bacterium. These phosphorus isotopes are present inside bacterium. It means from virus something enters or passes to bacterium. What thing was that which passes to bacterium? That thing is phosphorus. But phosphorus binds with DNA because phosphorus is component of DNA. So phosphorus isotopes are present in DNA. As this phosphorus is detecting and present or find out in bacterium, it indicates that DNA comes in bacterium from that virus. Here in this region, in this bacterium, no sulfur isotopes are present. All sulfur isotopes are present only and only in this protein shell or capsule. So sulfur is component of protein and hence sulfur isotopes detected in protein region, and protein is the material which is not supposed to be passed from virus to bacterium. Hence protein is not here in this bacterium. Isotopes of sulfur is not present in the bacterium. It indicates that protein is not transforming material. But as phosphorus is present in DNA and phosphorus is detecting in bacterium, it indicates that DNA transfer from virus to bacterium, and DNA is only the transforming material, as well as DNA is only the genetic material or heredity material which transfer from one organism to another organism. And in this way, Hershey and Chase prove that DNA is a genetic material. In the next class, we will discuss about RNA and comparative study about RNA and DNA.